For the growth and reproduction of organisms, cells must divide. Although mitosis and meiosis are both processes by which cells divide, they have very different outcomes. In this presentation, we used various types of candy to recreate the steps of both mitosis and meiosis.
Cell division is the important process by which living organisms are kept alive. Interphase is the longest phase of the cell cycle where the genetic material is grouped together in the nuclear envelope. This is followed by mitosis, which includes the phases of prophase, metaphase, anaphase, telophase, and cytokinesis.
Mitosis allows for cells to reproduce and regenerate tissue in the body. It is also important in its ability to allow for asexual reproduction.
During prophase, the chromatin condenses into chromosomes and the nuclear membrane breaks down. Meanwhile, the centrioles, which form microtubules, move to opposite poles.
In the following phase, metaphase, a mitotic spindle is formed from the centrioles and attached to the kinetochores, which are formed on the centromeres of chromosomes.
In anaphase, kinetochores are pulled away by motor molecules which travel on the microtubules and separate the chromosomes into two separate chromatids with their own centromeres, thus becoming chromosomes themselves. By the end of this phase, there are twice as many chromosomes as
In the
before.
next phase, telophase, all the chromosomes gather at opposite poles and separate nuclear membranes are formed around them. Cytokinesis is the process by which the cytoplasm is separated towards the poles. At this point, microfilaments are deposited on the metaphase plate and contract to create the cleavage furrow and eventually join the plasma membrane on either side.
Mitosis divides all somatic cells which sustain life. It consists of one cell division, resulting in two diploid daughter cells, both identical to the parent, and each has 46 chromosomes.
Meiosis is the process that makes sexual reproduction possible. Without meiosis, there would be no genetic variation.
Meiosis I separates homologous chromosomes in stages. In prophase I, the homologous chromosomes loosely pair up. Corresponding segments of DNA are exchanged through the chiasmata, while proteins hold the chromosomes together. This is known as synapsis, which does not occur in mitosis. As in mitosis, the chromosomes move, the spindle forms, and the nuclear envelope breaks
In
down.
metaphase I, the pairs of homologous chromosomes now become arranged on the metaphase plate with one of them from each pair facing each pole. Each homologous chromosome is attached to the microtubules from their respective poles.
Anaphase I is the process where homologous chromosomes separate because the proteins holding them together break down. The now separated homologous chromosomes move towards opposite poles with the guidance of the spindle. Sister chromatids remain attached at the centromere and travel towards the pole as a unit.
Telophase I is the phase in which each half of the cell has a complete haploid set of replicated chromosomes. Each of these chromosomes is composed of two sister chromatids in which one or both of these regions include non-sister chromatid DNA.
Through this and cytokinesis, two haploid daughter cells are formed. Meiosis has a second part that separates the sister chromatids.
In prophase II, a spindle is formed and the chromosomes move toward the center of the metaphase plate.
Metaphase II is where the chromosomes are lined up along the metaphase plate and are attached to the microtubules extending from the poles. Because DNA was crossed over during meiosis I, the two sister chromatids of each chromosome are not genetically identical.
Anaphase II is where the sister chromatids separate due to the breakdown of the proteins that hold them together. The chromatids then move toward opposite poles as individual chromosomes.
Telophase II is the phase in which nuclei form and the chromosomes begin to condense as the cytokinesis, division of the cytoplasm, occurs.
Meiosis reduces the number of sets of chromosomes from 2 to 1 in the gametes, meaning from diploid to haploid. This occurs in germ cells during the production of gametes. The two divisions of meiosis result in 4 daughter cells, which are distinct from each other and from the parent cell. Each of these daughter cells contains 23 chromosomes.